environmental conditions on a genome. The changes are going to be the chromatin. It's important to understand that DNA will not be changed. The environmental conditions that can affect chromatin are various factors in the environment, including air, food, water, country, culture, individual conditions, and these are going to affect individual cells, gametes, embryos. Epigenetics is important for cell differentiation since it's going to regulate gene expression in daughter cells. And it's also going to be important in genomic imprinting, which is the explanation why different methylated genes are expressed in different siblings and why no two siblings are completely alike. There are three molecular processes involved in epigenetics. The first one is DNA methylation. For example, if we have an ATCG sequence, a methyl group is going to be added to a cytosine on a CG or CNG islands. Cytosine is no longer going to be cytosine, it's now going to be methylcytosine. So when an old strand of DNA is methylated after DNA replication, it's going to be hemimethylated. When it undergoes DNA replication again, methyltransferases are going to come to the new unmethylated DNA strand and are going to methylate the cytosine groups in the CG or CNG islands to make identical to the old strand. DNA methylation is going to inhibit transcription, inhibit binding proteins, attract transcription repressor proteins, and histone deacetylase enzymes, and this in turn is going to inhibit gene expression and elicit phenotypic changes. Another molecular process involved in epigenetics is histone modifications, which in turn is divided into two. Acetylation, which also involves deacetylation, and methylation, which also involves demethylation. Acetylation is when the enzyme acetyltransferase transfer an acetyl group to a positively charged amino acid, lysine or arginine, on the histone. This is going to neutralize the positive charge of the lysine or arginine, and this is going to loosen the grip that the histone has on the DNA, thus relaxing the DNA, promoting transcription, turning on genes, and increasing gene expression. One example we have is the repressor gene FLC, which is going to be responsible for the flowering or non-flowering of plants. And this in turn is going to be influenced by climate and environmental conditions. For example, in the winter, histone acetylation occurs in this gene, and this gene is expressed due to the low sunlight conditions and harsh climate conditions. However, in spring, when conditions change, there will be deacetylation, which is going to stop FLC expression and allow flowering to occur. There will also be methylation involved, which, as we said previously, is the addition of a methyl group, which can increase or decrease transcription. Temperature can also influence demethylation of the DMRT1 genes in turtle and the KDM6B genes, which are going to influence if the turtle are going to be females or males. Lastly, we have the effects of RNA molecules, and for this we need to understand the term mutation, which is the interaction between two alleles, and it's part of a post-translational modification. The effects of RNA molecules can be on long non-coding RNA, siRNA, miRNA, or piRNA. Now we're going to mention some examples. The first example is honeybees. The function and size of honeybees are going to be determined by what kind of diet they have. For example, queen honeybees eat royal jelly, which silences the DNMT3 gene, which is going to reduce the likelihood of DNA methylation. However, worker bees are going to eat common food, which is going to cause DNA methylation, and this common food is pollen and honey. And there's also going to be miRNAs involved, which are going to suppress the fertility and genes development. Example 2 is the naked mole rat, which is going to involve DNA methylation and histone modifications. Since the lower-ranking females in a naked mole rat population are not going to develop ovaries due to their diet, which is the poop pellets rich in estradiol of the queen naked mole rat, which is going to activate the maternal instinct in the lower-ranking females, so they can raise the queen's offspring, but also going to affect their own fertility. Example 3 is the AVY mice, the agouti viable yellow mice, which is normally obese and yellow, and that's what makes it viable. However, when they are fed methyl donor diets, which are rich in folic acid, vitamin B12, betaine, choline, or soy phytoestrogen, there's going to be methylation that occurs, which in turn is going to affect metabolism and color and give us a lean brown color mice. However, male parents can undergo effects of RNA molecules causing epigenetic changes. If they have an environment rich in physical exercise and mental stimulation, they're going to be positively influenced as RNAs, which are going to in turn lead to better brain development and memory function in their offspring for this population, for this species of mice. Another example of the effects of RNA molecules is the X chromosome inactivation in humans. Since the XIST gene is going to influence the long non-coding RNA, which is going to attach to one of the X chromosomes and inactivate it by making it heavily methylated. This paramutation also occurs in breeding corn. When you breed a purple corn with a green corn, the green corn, the siRNAs of the green corn are going to silence the genes, the alleles of the purple corn. And all of this, DNA methylation, histone modification, and effects of RNA molecules are going to be greatly involved in behavioral epigenetics. For example, maternal effects have an influence of on DNA methylation and histone modifications. And we see positive effects such as less fearful offspring and less stressed offspring in mice, depending on where the mice spent their lives, whether it was in a laboratory setting or a more free setting. Stress is going to lead to depressive behavior, underestimation of risk, and abnormal miRNA in sperms in mice. Effects of RNA molecules are also seen in the human population. Small non-coding RNA differences can be seen in adults with post-traumatic stress disorder. Also, childhood abuse is an example of DNA methylation. Since there will be an increase of methylation of the glucocorticoid receptor gene and or shortened telomere length, which is going to lead to behavioral abnormalities in these populations. Poverty can also increase methylation of the SLC6A4 gene, which encodes a protein that transports serotonin. If no serotonin is being transported, there will be more likelihood of depression, and this also leads to an overactive amygdala. However, exercise decreases DNA methylation of bad genes and increases DNA methylation of good genes, which can enhance gene expression for muscle remodeling and cellular genetics. Has also been proven effective in mice populations since there will be a normal methylation of the PCG1 gene, which is going to lead to normal glucose and insulin levels. Also, smaller dogs live longer due to DNA methylation of cytosines, which lead to longer lives. DNA methylation and histone modification also occur in monozygotic twins. That's why even monozygotic twins are not similar behaviorally. As they grow up, based on their individual circumstances and experiences, there will be an epigenetic drift. Okay, that is